first of all, about the rap lyric Google Doc group collaboration assignment, where it was clear that my expectations weren't made clear by me for all of the groups. So now that I've posted the Blackboard announcement about, which I think clarified my expectations, hopefully that will improve things for the next time we do this exact same exercise at the end of the upcoming chapter. To be really clear, what I mean is, the idea is that everybody uses the Google Doc that's shared among your group, and every individual uses Google Docs, the app, to access that doc, to edit it. Because I can see who contributes what to each shared Google Doc, then I can assign points accordingly, which basically is, did you participate or did you not? And this time, everybody's going to get the 1 point, because some of you didn't, because I didn't communicate, and so some of you didn't understand what I was expecting. And so when I got screenshots of Google Docs or PDFs of Google Docs, I can't see the revision history, and so I have no way of knowing who contributed what. So this time only, everybody's going to get the 1 point. Future assignments, future chapters where we do the exact same thing, everybody has to use Google Docs to edit the document. The group leader has to post the Google Doc to Google Classroom, not some other version of the Google Doc, like a PDF or a screenshot. Okay? So that's moving forward. For now, everybody gets the point. Next chapter, now my expectations hopefully are clear. Any questions about the whole Google Doc fiasco? Yes?
Is this something that we can ask you about? Because our group tried a couple of times and we still can't get it.
Yeah, of course.
So go to like a tech center or something and ask them. I don't want to like flood you with people. Right.
They did several things that just didn't work.
Yeah, it's okay to ask me. It's okay to ask our Discovery Guide on the Go that's in the class. It's okay to ask the Discovery Hub.
Okay.
Yeah. But I do want to make sure, and the reason that I'm really pushing the collaborative Google Doc process is because we might wind up using that on an exam, like the midterm. Because if I hadn't said it before, exams will also be taken on your devices. They will most likely be open note, open internet, open everything. So we've got these in class, we might as well use them on exams. And so I want to make sure that everybody understands how to do PDF annotation, collaborative Google Doc editing, and attaching to Google Classroom, because that's the workflow for the exams. That's how I collect what I'm going to grade for your exams. So I need everybody to actively work, and that's why we're doing all of these exercises in class, to deliberately work that direction, so that everybody can do this in class on the day of the midterm and successfully complete everything by the end of the 50 minutes, so that you're not freaking out about, wait, now my tablet can't do this thing that I never practiced. This is practice. Other questions, comments, concerns? Yeah.
Okay, so
Clarifications?
With the Google Docs, I noticed that whoever opens it is the owner of it, and then everybody else edits it. So if we're one of the people that's just one that can edit it, we can actually send you the copy of the Google Doc, and that actually does show the comments and revisions on there.
So the person that creates the Google Doc is the person that should attach it to Google Classroom. Other concerns, clarifications, questions? Okay. Please.
Is that a rule that if whoever created the doc has to upload it to the classroom, or can someone else do it?
That's the way that I can see the revision history. So if somebody else posts it, I don't think that I see the revision history. That is who contributed what. Delete things, add text, edit text. So I prefer it if the person that creates it also be the one that submits it.
Well, that's the only one that can really submit it because they own the doc.
That's right. That's right.
So we're not required, the people that are in the group that didn't open it aren't required to submit it, or you still want us to submit a copy?
The reason that I'm asking for the file name and the contents of the document to include all the names of the group members is so that when the one group member that owns it submits it to me, then I give all the group members that are listed in the document title and in the document text the points. Yeah, so only the group leader, whatever you want to call it, has to submit back to Google Classroom. And if you don't have your point in Blackboard yet, it will be coming within the next 24 hours. If it doesn't, let me know. Thank you for helping me clarify this, because I want to make this easy. I don't want to make the technology a burden, but I do want to make sure that everybody understands how to do this. So I don't want midterm day to be as stressful as it will be if you're taking an exam plus you're trying to learn new things on the tablet all of a sudden.
Could you repost the websites for the videos? Because I changed it and it still wasn't. I wasn't accessed.
The videos for today? Okay, so I'll post those somewhere else. Okay. Folks in the back, questions, concerns? Any questions, comments? Okay. What did we talk about last class? What were the key points? What do you remember? What did we talk about?
Adaptation,
Yeah, 2 classes ago was adaptation versus exaptation. That's just demonstrating really good memory. Oh, we talked about, right, so we talked about C. elegans, the evolution of hermaphroditism, and then there was one other case study we looked at. The LTEE. This is not some new network that Verizon runs. It's the long-term evolution. I don't know. Anyway. It's the long-term evolution experiment from Rich Lenski. So that was bacteria. Rich Lenski's lab, that is. Him plus whoever's worked in his lab over the last couple decades. Okay. So what was the point of that whole class? If you summed it all down to one key point about evolution.
That it's an accumulation of mutations over time before you really get the mutation that makes the difference.
So there are some times when you have to have a bunch of mutations, all of which together eventually give you some adaptive trait. So it's not necessarily the case that there's just a single mutation that has to occur to get you to being more fit than you were before. And that was true in both of these cases. There are a series of mutations that have to happen, and each of those mutations by itself doesn't change your fitness. So a lot of those mutations are what we call neutral mutations. They're not good, they're not bad, they're just DNA changes in your genome. But then when that last one arrives, all of a sudden, all together, this group of mutations confers some either advantage or disadvantage, and it's selected for or against. Evolution's not usually easy. So today we're going to cover a couple of concerns or clarifications about natural selection, things that people commonly ask about. Wait a second, if natural selection works this way, then why, for example, don't organisms always seem to be optimally adapted? So this is a huge concern among people that debate, does evolution occur, does it not? Does natural selection occur, does it not? So what about peafowl? Does that male seem optimally adapted? So if you say no, why is this non-optimal?
It's optimal for him to reproduce, but it's not optimal for him to live in his environment.
So it's great for reproduction, she says, but not for living in the environment. So how could the huge tail be maladaptive? How could it be bad? It takes a lot of energy to produce, so at the cellular level, the male had to invest a lot of metabolic resources into producing it in the first place. Then it takes energy to cart the whole thing around. And likewise, it's flashy, it's bright, and so it's likely to attract, as you said, predators. So there's always a trade-off, which we've discussed before in class. So the trade-off might be, well, yeah, you're more likely to get killed by a predator who spots your tail, but at the same time, why is this a beneficial trait? As was said up here already, it's for mating. Right? There's fitness involves surviving to reproduce, but then actually being able to reproduce. So there's the two components. And presumably, the peacock's tail strikes a balance. Probably not great for survival, but great for reproduction. What about antlers? Cost or benefit? Great, yes, trade-offs. So why do, if you had to pick a reason, so maybe adaptation or exaptation, I'm not sure, why do deer, elk, moose, et cetera, have antlers? Okay, so defense against other predators. What else? And for
They use them for the mates.
right, so they're part of they might be part of mating. So what do, if you Google deer antlers, what sort of pictures are you gonna find? Well, right, you're gonna find pictures of deer antlers. You're gonna find stuffed deer heads.
Only like different sizes, probably.
Then you're gonna see pictures of two male deer fighting, right, for a mate. So it's all about, right, antlers usually, again, is this the adaptation or is this an exaptation? Did the antlers evolve and then they started using them to combat other males for access to mates? Or did the antlers evolve in the first place for competing for mates? We're not sure. But is it maladaptive? In this case, it seems like maybe antlers are sometimes not very useful if they get stuck in a tree and the hunter has to come, I don't know what's going on. The hunter is actually going to go rescue the deer. Like the beneficial, the kind hunter, the pacifist hunter. But it looks like they're trying to get this buck out of this tree. So maybe not optimally adapted, but certainly there's a benefit to having the antlers, otherwise they would probably be selected against. Please, yeah.
Is this similar to what Darwin says is nature being wasteful, or is that something else?
So is this related to Darwin's concept of nature being wasteful? So can you give a little bit more context for
I
that?
think in the second chapter, he talks about, in Origins of Species, he talks about how nature could be wasteful. Is this applying to it, or is it more like if a trait makes an organism become extinct, it becomes wasteful?
So how does this apply to nature being wasteful? I'm not sure that this is what Darwin was thinking about. I don't know though, of course, that's just my opinion about the wastefulness of the economy of nature. But let's talk more about it after class if you want to pursue this, because I'd want to know what you're getting at. So there are other reasons that organisms aren't usually often optimally adapted. So, of course I didn't create this, just something I found on the internet. I'm not trying to offend anybody, but it is generally, you've heard this before, it's generally a rule that inbreeding is not a good idea. That's for a few reasons. So one reason that organisms might not be optimally adapted, and this goes back to something we talked about last class, maybe a few classes ago as well, is what does this have to do with? Why don't we always see optimal adaptations in organisms?
It's because it's a random mutation and control.
So random mutations allow adaptation to your current environment. You don't control where the mutations happen, how frequently the mutations happen, nor do you necessarily control who you mate with. We can, most organisms can, but some organisms don't. And the smaller your population, the smaller number of people you can choose from, the worse off you are. So a smaller population size, and this is a topic we'll talk about again and again in class, which it says up there, reduced variation population bottlenecks. When you have a very small population of individuals you can choose to mate with, that means you have less mutation. Fewer people, fewer genomes, fewer times your chromosomes replicate, fewer mutations. So small population, where does this apply in modern day? I mean, it applies to humans, sure. What's an application of this concept to concerns in modern day biology?
Endangered species.
Yeah, endangered species. So when you have a species that has really low population sizes, they're worse off, and they're worse off, and they're worse off. It's a snowball effect. The fewer individuals there are, the more inbreeding there is. The more inbreeding there is, the less genetic variation there is. If there's less variation, then that's one of the critical components of natural selection. You've got to have variation. And if there's less variation, because there are fewer individuals, you're less able to adapt. Natural selection doesn't work as well. So reduced variation leads to reduced natural selection. So natural selection still happens, it's just less efficient. So one thing we might talk about in class later in the term would be how do you best help those endangered species when there are small populations? What are the practices that zoos and other conservation groups put into place to try to help, as best as we understand genetics and natural selection, help maintain diversity in those species? And related to that is this final point, which we've also mentioned briefly before. What's optimal is usually a moving target. It depends on where you are in the world. It depends on the current climate conditions. It depends on a lot of things. What if you're a polar bear? So what happens, what's the gestation time, what's the generation time of a polar bear? I don't know. Somebody Google it. Few years, maybe. But generation time, so how old does a polar bear have to be to reproduce? I don't know, you can find out for me maybe. Right, the longer your generation time, think elephants. The longer your generation time and the fewer offspring you have, the smaller your population. If your population is small and what's optimal is changing for you because of global warming, say, then how is your species ever going to adapt now to conditions now when you have to wait another 10, 20 years to have the next generation to find out whether or not there are any new mutations in that generation that may or may not be better suited to that climate. By then, the climate might have changed again. pH in the water changes, global temperatures change, the type of predator in your environment changes, the preference of females in that environment changes from peacocks with eye spots on their tails to peacocks with stripy tails. I'm just making stuff up now. But that's what happens. Female preferences change, so the males are always trying to make whatever the females like, so to speak. Not consciously, of course. But that's how natural selection works.
It's 2 to 3 months.
Pardon?
It's 2 to 3 months gestation period.
Oh, wow. That's pretty fast. 2 to 3 months gestation.
Generation time,
It's 11 and a half.
The generation time is 11.
Okay, so then 11 years. So they have to be 11 before they're
Yeah.
reproductively mature.
But the gestation period is really short.
But short gestation period, so they might be able to have a lot of offspring. Okay, so I'll stop using polar bears as the prime example of long gestation times. You learn something new every day. Okay. So any questions at all about these sort of mini case studies? So natural selection requires variation. Population size is reduced, variation is usually reduced, and that's usually, not always, a bad thing for natural selection. We don't see organisms that look optimally adapted, sometimes because there is no way to become optimally adapted to that situation. Moving target, in other words. And then as we're going to see next, and for the rest of today's class, we're going to talk about coevolution or arms races, something that some of you mentioned a few classes ago you wanted to know more about. I mentioned this term. So what is an evolutionary arms race? And we're going to look at a couple of examples. One, we're going to talk about antibiotics. And then the second, we're going to talk about host virus coevolution, immunity. So these are situations where natural selection is almost powerless to improve the situation of either individual that are part of this tug of war, the arms race. Have you ever heard of the term arms race outside of this class? So you're right, Cold War is usually one of the first things people mention. So Cold War, Soviet, back in the day, Soviet Union and America, there's escalation. So one side gets nuclear weapons, the other side has to have nuclear weapons to deter the threat, and then the other side says, well, okay, we need something better than nuclear weapons, so there's that back and forth. That's the arms race idea, that you're always trying to react to the second party. There's always two parties, two groups in an arms race. You're trying to outcompete each other for dominance. And in evolution, usually arms races don't have winners. It's just a constant dynamic of one-upmanship. Oh, you did that? Okay, well, I'll do this. Back and forth, back and forth. So this is pretty clearly the case in the case of antibiotic resistance in bacteria. So you can look at the details thoroughly by yourself later if you want. But this is sort of a brief timeline of when different antibiotics were introduced in history, and then the points in time when bacterial resistance evolved. So you can see for every one of the antibiotics that were introduced on the top of the timeline, at some point, a decade or two, maybe later, the first cases of resistance in bacteria to that antibiotic were recorded. So this is sort of step one of the arms race. You apply antibiotics, bacteria evolve resistance. So let's look at a few reasons why the Cold War in bacteria happens in the first place, or how it happens. So there are a few ways that bacteria are vulnerable. So these are their soft spots. This is the underbelly we're going to try to strike. So bacteria have high internal osmotic pressure, and so they have to have strong cell walls to keep themselves from lysing, from bursting. So it's a weak spot in their defenses. If we can attack the cell wall, then bacteria rupture and we kill them with antibiotics. So that's one way we might attack the bacteria, attack their cell walls. Another place that bacteria are vulnerable is in the way they replicate their DNA. So with circular genomes, when the circular genome gets replicated, there are some enzymes the bacteria have to use, like topoisomerase and gyrase, to name a couple. Because their genomes sometimes get concatenated or linked together when that circular chromosome replicates. It's like the magician's trick. You've got two linked rings, and the magician somehow pulls them apart and unlinks them. So bacteria have enzymes to do the same thing. In the case when they replicate their circular genome, those two genomes are linked together and can't come apart. These enzymes release them so that they're separate circular genomes. So this is another place where they're vulnerable. We have antibiotics that prevent these enzymes from working, and when the enzymes can't separate or decatenate the circular chromosomes, then the bacteria can't divide and they're essentially dead. If they can't divide, they're not a threat anymore. And of course, as with us as well, bacterial ribosomes have to work to produce proteins. you can't make new proteins, then you're basically useless as an organism. So this is another place that we attack bacteria with antibiotics, is prevent their ribosomes from synthesizing new proteins. There's three ways we attack bacteria with antibiotics: the cell wall, DNA replication, and protein synthesis. We target those three essential functions. Where do our antibiotics come from that we use against bacteria? Right. Does it say that up there? Ah, yes. Good job. So, right, this is actually where the arms race is. It's not us treating infections with antibiotics. The bacteria were the ones that came up with the antibiotics in the first place. So what is this picture showing on the right there? This is a famous experiment, if not a picture from the original experiment.
Showing antibiotic resistance.
So we've got, it's showing antibiotic resistance. What's that big fuzzy thing, do you think? It's penicillin. It's penicillium mold. I hope I can spell that correctly. Right? That was that fortuitous experiment where somebody presumably left the petri dish out and some mold floated in the window and landed on the petri dish. So this is kind of a recreation of that. And when this penicillin mold grows near, what bacterium is this? Don't remember. I'll remember by next class, I'm sure. It produces this chemical that causes this clearing. Right? It actively kills bacteria that are near it. And so Fleming said, oh, look, it's like this mold is producing something that's actively killing bacteria. What's happening over here? There's a penicillium colony in the middle. There was a clearing. So what are all those little spots in there? So those are bacterial colonies of that original bacterial culture that was on the plate that have become resistant. So now they can grow in that ring that originally was killed by penicillin. Now there are resistant colonies starting to reinvade. So here's the arms race in action. We had penicillin that has evolved to produce something that kills bacteria. Now we have bacteria evolving to overcome whatever the effects of penicillin are to recolonize this territory. So what's the next move in the arms race, do you think? So it's penicillin's turn. So it's going to, presumably evolution will help it evolve a stronger version of penicillin or a different antibiotic. Who knows? But that's the concept of the arms race. It's that back and forth. I'm not sure why I left this slide in here, but so chemicals are not antibiotics. This is just to reinforce that idea that antibiotics are produced by organisms, which means that they evolved. Antibiotics, that is. The ability for an organism to produce a chemical that kills other organisms is one of the steps in the arms race. It involves evolution. This text is pretty small, and it's not crucial. I just wanted you to see, you can look at this later if you like, where some of our modern antibiotics target those vulnerable aspects of bacteria. They attack the cell wall, they inhibit topoisomerase or gyrase, or they inhibit the ribosome. So most of our common antibiotics we use now do one of those three things. So if you have which some of you probably do right now, I'm kind of feeling like I might have an infection. If you've got an infection, I don't want to put my own medical opinion on you. When I go to the doctor, though, this is my story. When they try to prescribe antibiotics, I ask a hell of a lot of questions about why. Because what happens if a doctor prescribes me an antibiotic and it's not an antibiotic that will kill the type of bacterium that I'm infected with?
The bacteria becomes resistant.
So it will kill some of the bacteria maybe, but if those bacteria are resistant, I'm actually selecting in my body for more resistant bacteria. So this is why bacterial resistance is so, or antibiotic resistance is so common now, is because of a long period in human history of widespread antibiotic use. So we're always, by using, and again, this is my opinion, by using the antibacterial soaps and cleaners and so forth, we're just providing more pressure to select for bacteria in the environment that were resistant to all that chemical and antibiotic that we throw at the organisms. So that's the next step in the arms race. We widely apply antibiotics, and the bacteria have a better advantage if they happen to have those random mutations that make them not sensitive, resistant. Now, this whole story about bacterial antibiotic resistance, I want to make really clear, is very different from the summary of last class that we heard earlier, which was that it often takes a number of mutations to go from one evolutionary state to another, to an advantageous one, to gain a new trait that's beneficial. So that's the reason I'm showing you this table, which is a small table of a few different antibiotics here on the left side, the names of the drugs. And DNA positions, so regions, so SS rRNA is a small subunit of the ribosomal RNA, for example. So streptomycin at position 523, I'm just reading across the top line here for an example. So streptomycin does something that involves action at the 523rd nucleotide in the SS ribosomal RNA gene. Eubacteria, the ones that we're usually trying to kill with antibiotics, are sensitive. So the S in the susceptibility column means they're sensitive to streptomycin if they have an A at position 523. Chloroplasts, a different type of microorganism, are also susceptible. They also have an A at that position in that gene. Right? So we're talking about nucleotides here. Single nucleotide, one position in this gene, the ribosomal RNA gene. What does it take to become resistant to that drug? If you keep reading across that line. So this is a weird case. So interestingly, in eukaryotes, an A causes resistance. So this is kind of a weird situation. But mitochondria have a different nucleotide there. No longer an A, now we've got a mutation. Our mitochondria have a C at this spot. They're also susceptible to this drug. So this is probably a case, this is confusing. This is probably one of those cases where there are multiple mutations necessary to make you susceptible, make a bacterium or mitochondria or chloroplast susceptible or resistant to a drug like streptomycin. But now look at the one on the bottom, the macrolides. This is a much clearer case. So what's the pattern? If you have a particular nucleotide at one spot in one gene, if you're a bacterium or a mitochondria or a chloroplast, you look in one gene, one specific nucleotide. How do you go from being susceptible to this antibiotic, a macrolide antibiotic, to being resistant? Yeah, so if you're susceptible, you always have an A at that particular spot in the genome. What does it take to become resistant? One mutation. All you have to change in your genome if you're a bacteria is have one point mutation from A to G, and you become resistant. So this is not like the earlier cases where it takes a lot of mutations to develop an adaptive trait. If you're a bacterium and you happen to get this one point mutation, you go from being sensitive to a drug to being resistant to a drug. So it's pretty easy, especially given the huge population sizes of bacteria, how quickly they replicate, and how many mutations get made every time a bacterial genome gets replicated. Now, hopefully this is interesting from another point of view, which is that this is why we can use antibiotics to treat some infections and not kill ourselves. Because what is it that's resistant to this drug? Which type of bacteria that we have in all of our cells? What's resistant? Our mitochondria. Because mitochondria used to be bacteria. They are kind of bacteria. They're bacteria that, according to many hypotheses, are ancestral eukaryotic cells engulfed and enslaved to make our cellular energy, ATP. So they're bacteria. They have bacteria-like ribosomes. And so, of course, we want to have antibiotics that kill certain types of prokaryotes, like the bacteria on the left of the table, but we don't want these antibiotics to target our mitochondria. So it's beneficial for us that there is a difference between the sequence of DNA in our mitochondria that make them resistant to this drug and the sequences of other microorganisms that we're trying to kill with the same drug. It makes it rather difficult for chemical companies to come up with or to tweak to chemically engineer new antibiotics because they have to make sure that those antibiotics are only going to be killing the bacteria we're trying to kill and not harm our mitochondria.
Does streptomycin harm our mitochondria, or is it because the cytoplasmic is resistant, then it doesn't harm the mitochondria?
So presumably, and so this is a good question. So what about streptomycin, the top one? Why do we use it? Is it harmful to our mitochondria? I don't know the specific answer to that. My guess from a scientific standpoint is this is why it's important to know what dose you're taking. So you give enough dose to kill certain bacteria, but not enough that it gets into our cells and kills intracellular bacteria. Right? So another thing an antibiotic has to do to harm our mitochondria is get into the cell. So a lot of antibiotics, of course, are invading bacteria are not in our cells. They're in our bloodstreams, they're in our gut. So it's easier for the antibiotics to access them. So a lot of these antibiotics don't get into our cells to harm mitochondria. But if they could, that would be really bad. Other questions? So again, one case study that shows how easy it is in certain cases to evolve something that's beneficial. Single point mutation can confer resistance to certain drugs. Okay. So the other case study is about host-virus interactions. So we're the hosts. We're going to talk about poxviruses today. Chickenpox, smallpox, vaccinia viruses, for example. So here's a vaccinia virus. It has a protein called E3L. So I apologize for having to use the protein nomenclature. It's boring, I know, and it doesn't really mean anything. But in this case, poxviruses, they're double-stranded DNA viruses, which is weird for viruses, but it means that they replicate more slowly. So these are not viruses that have RNA genomes, they have DNA genomes. They have kind of a different lifestyle than most viruses we're used to thinking about. And these viruses all have a protein. They're called different things in different species of virus. So NSS, sigma-3, NS1, TSR1. We're talking about vaccinia, so we're talking about E3L. All of these proteins, indeed, have one function. All of them inhibit, right there, this protein called PKR. So all these different viruses have different proteins, they're related, but they all have one job, to inhibit a protein we produce called PKR, protein kinase R. So those of you that have taken biochemistry, what does a kinase do? Yeah, phosphorylates other proteins. So in this case, we have PKR and it's beneficial for us because it does a lot of things to help us stay healthy. So that's the whole point of this figure. You don't need to memorize the details. It's just to make one point, which is that PKR, shown at the center of the figure, does a lot of different jobs in our cells, and they're all to help us fight off viruses. So what's showing up here is that here's a virus, it infects a cell, here's its double-stranded DNA genome. Our cells have proteins that go around monitoring for the presence of double-stranded RNA, which is produced by viruses as part of their life cycle. So normally you know that RNA is single-stranded, and you only find it in its single-stranded form. It gets translated by ribosomes, makes proteins. Double-stranded DNA is usually, when found in a cell, is usually a sign that there's a viral infection. So our cells have machinery to go just always looking in our cytoplasms for double-stranded RNA. And PKR is one of the protein that responds to that detection. So here it's showing PKR binding to the double-stranded RNA. I'm not sure if it actually directly binds to the RNA. But one of the responses is shown here on the right. The protein kinase R pathway recognizes double-stranded RNA. It causes the cell to kill itself, apoptosis. If you're a virus, what do you not want to do when you infect a cell? Yeah, you don't want to. Yeah, you don't want to set off the cytoplasmic or the apoptosis machinery. You want the cell to live long enough for you to reproduce, and then of course when you reproduce, you kill the cell, you lyse the cell, and all of your progeny get released and go and infect other cells. But you don't want to trigger the protein kinase R response. Protein kinase R also has other jobs. Shown in the bottom half of the figure is basically saying that PKR turns on transcription factors that turn on genes that are also involved in antiviral response. So PKR is like the gatekeeper. When it senses a viral attack, it activates all of the cell's responses to, hey, we've got a virus in here. So how does the virus respond? This is an arms race. So the cell has PKR. These viruses have these proteins that recognize PKR and somehow inhibit it, turn it off, make it no longer able to function. That's two steps of the arms race. We have defense. The virus evolves, presumably, a protein that disables that defense. So whose turn is it now? Okay, so then it's our turn. So the virus is producing a protein that's inhibiting PKR. So what do we do in response in this arms race? Pardon? So, right, it selects for mutations in PKR. So this is the arms race. So imagine that initially our PKR has a particular shape and the virus, in this case the K3L protein, recognizes that specific shape of protein kinase R and turns it off. So what do? We have mutations in protein kinase R that, diagrammed here, change its shape. So if one of us happens to be infected with a pox virus, and we happen to be carrying a mutation that makes our protein kinase R protein unable to be detected by K3L, then we have an advantage. We're not susceptible to the pox virus. So we can't turn off our cellular defenses, so protein kinase R kills all the cells that have this infection. We're healthy, we go on to reproduce, we have lots of kids, pass our genes on, everybody's happy. So then how does the pox virus respond in this arms race? Pardon? Right, so then that's the arms race. So then at some point, if a pox virus happens to infect one of these people that's resistant, if that pox virus happens to be carrying one of these random mutations that makes its K3L able to recognize the new shape of PKR, then we're back to square one. It's that back and forth tug of war. Nobody wins. It's always one side's turn or the other to have that evolutionary response to the conflict. That's why this is called, in case you were wondering, although no one's asked yet, maybe because I'm talking too much, the Red Queen conflict. Why this is called a Red Queen conflict. It's just another term for an arms race. If you happen to have watched Alice in Wonderland, that's where this term comes from, the Red Queen conflict. At some point in Alice in Wonderland, Alice is running and running and running and running and running and running and not getting anywhere. And mentions to the Red Queen, hey, why are we working so hard and we don't seem to be getting anywhere? And the Red Queen replies something to the effect of, all you can do is put as much effort in as you can to stay in the same place, or something like that. It's an analogy for the conflict, that both sides of an arms race are putting a lot of effort into maintaining the status quo. Because no one ever wins. It's just trying to win. Both sides are evolutionarily trying to beat the other side, but there's never any advance in the war. It's always a tie, in essence. Sure, if you give up, then you're going to lose, and evolution never gives up. So it's just constant back and forth coevolution. One side evolves, the other side responds, and so forth. And the reason I'm telling you this story is also because, remember I said that this virus has a double-stranded DNA genome, and it replicates a lot more slowly than RNA viruses. So these researchers that were working on this particular study were curious how a DNA virus evolves so quickly to respond to changes in our PKR gene. Because it's clear that it's not just the sheer number of offspring that a virus has, and relying on pure mutation to drive adaptation, to produce the variants that can be selected for. So what they discovered was that K3L gene in the virus, there are numbers of copies of this gene in the viral genome. So it's not just that there's one gene in the virus that produces that protein. There are lots of different versions. There are lots of different copies of that one gene, which increases the chance that every time the genome is replicated, a random mutation happens in one of those many copies of that gene. It improves their chance. There's a bigger target, in essence, for natural selection. More likely that every time this genome replicates, that one of those 5 copies, say, gets that random mutation that confers an advantage. And they also discovered then that after one of those mutations arises, that over time the viral genome moves from that state of having multiple copies of the K3L gene back to having only one copy. So the viral genome starts out with lots of copies of this gene, but once it's got the one version that helps it win the war against PKR, it gets rid of the rest by natural selection. So it's expensive to keep these multiple copies of the gene around. Every time you replicate the genome, you have to replicate all of those genes. It takes a lot of nucleotides. So over time, that one copy of the gene that is the best at defending the virus against PKR gets retained in the genome. The rest get disposed by natural selection. Okay. So we've got a half a minute left, so I'm going to skip, although we're going to come back to this later in class anyway. An example of coevolution arms race using HIV as an example. You might have seen, so 2 more points. One, you might have seen that I passed out a Google Classroom assignment. It just has a link to a survey. It's only a survey with basically 1 question. It asks you your name, and then there's 2 questions, I think. So it's a very short survey. Please take it by next class. I think the assignment says it's due today. I'd appreciate it if you would. Really fast, 5 minutes or less. If you did bring money to contribute to the wrap thing, and you didn't give it to me at the start of class, I'll stick around here to collect now if you want, or on Monday if you didn't bring funds today. And for next class, the only thing you have to do is read 2 sections, chapter 4, 4.1 and 4.2. Everybody have a fabulous weekend, and I'll see you next week.